cool. Record, and we'll see how this goes. So before we really start, does anyone have any like general questions about how the test is going to go, or cheat sheet questions, anything like that? Okay. So I think at my last session we kind of discussed different like ways to format the cheat sheet. Like I used Times New Roman size 4 font. I used a lot of tables.
What did you print this on, a laser or inkjet?
I just had my regular like printer.
Yeah.
I don't know.
But this is size 4
Inkjet.
times 0 because I can
Yeah.
do this.
Yeah. Also, I think with the borders, you can do like 0.25. It's like on the Word document, you can do like 0.13 inches, but that's not actually going to print on like your standard printer. So like 0.25 is like a safe bet for what you can do for the outer borders. And then like other things you can just write in with that extra 0.25. Let's see. Use colors sparingly, I think, like only for dividing sections. Be familiar with your cheat sheet and try to group things together as best as you can. Try to group the clinical cases with like the relevant topics. I think I usually keep them in the blocks that he presented them on in his website, like all the infectious disease together, all the endocrine together, that kind of thing. And then yeah, that's pretty much it. Okay, so we'll just kind of go through some of the PowerPoint slides that I picked out, and then see how that goes. So to start off with hypoxic injury induced by ischemia, I threw this on my cheat sheet just because I felt like these kind of things, like a lot of pathways, a lot of possible things that you may or may not remember. Like you might not remember if something is like reversible or irreversible, and it's just general like information that like I thought may or may not have been helpful. Necrosis, it's important to know like the different kinds. So coagulative is like, a kidney could have an infarction, but also you could have with the liquefactive, and with the liquefactive, it does have an abscess for the necrosis, and that's kind of like the key word if it shows up on the exam. Caseous necrosis, think tuberculosis. Fat necrosis is with saponification, and gangrene. That's with your Clostridium perfringens, and diabetes also can get these kind of problems. Moving on to genetics. So remember with genetics, you've got your DNA code that covers all of the genetic information to make proteins. So from DNA, it goes to RNA, and from RNA, it goes to proteins. And proteins can be like your enzymes, your receptors, anything that like for structural components, that kind of thing. For these genetic disorders, make sure you guys know the difference between dominant versus recessive, and the difference between autosomal and sex-linked. So remember with sex-linked characteristics, it's inherited on, usually it's an X-linked disorder if it's a sex-linked one. And if it's autosomal, that's like all 22 other pairs of chromosomes. So it could be like chromosome 1 carries this disease, chromosome 2, 3, all the way up to 22. Whereas sex-linked only deals with the X and the Y, the 23rd pair. And if he does ask you a question about like Punnett squares and stuff, always just draw it out, try to map it out, and then you have like the little squares that you draw. Does anyone need to go over like how to draw a Punnett square or anything like that? And then I would know, you guys don't need to know like what these 3 syndromes do, but know the difference between aneuploidy and polyploidy. So aneuploidy is having like an extra chromosome or less chromosome, but polyploidy is like an entire set. So then Down syndromes, Edwards, and Patho's, they're like the 3 non-sex, like so they're the autosomal trisomies, so 13, 18, 21. And then Turner's 45X is the only monosomy. And then Klinefelter's is the sex-linked trisomy with 47XXY. Remember, with non-disjunction, this can occur in either meiosis 1 or meiosis 2. So that's when you've got your entire either homologous pair. The tetrad is what it's called when it's both homologous, like from the mom and from the dad. They can group together into, like if you see here, they group together in this one, or they get grouped together like none at all for here. But you could have non-disjunction in meiosis too, where the sister chromatids will segregate out into one versus the other. So be sure to know the difference between chromatid, chromatin, chromosome, centromere, all of those like C kind of words. So to review, with a chromosome, that's the maximally condensed genetic information during these meiosis, mitosis replications. Chromatid is kind of like is like each one of these like lines, so it's 1/2 of the X, you could think of it. So when you duplicate the DNA in S phase for the chromatids, like it'll kind of piece together to be these, it'll be duplicate DNA, so it'll be like that X that can form. So each individual half of a chromosome, is the chromatid, and then these two together is a homologous pair, so like chromosome 1 with chromosome 1. And then this, in the beginning, that's where you can have homologous recombination. So that's like exchanging genetic information between like mom and dad's pairs to create some sort of new diversity. But then if you had like chromosome 1 with chromosome 5, for example, that would be considered a translocation. So non-homologous chromosomes exchanging genetic information. Does that make sense? So homologous
There's a lot of like on this market says like non-homologous balanced and
Right.
homologous balanced.
Right. So like non-homologous balanced would be the translocation. Homologous but balanced is just a regular recombination, and then or crossing over kind of thing. And then so crossing over is normal for like chromosome 1 and 1 or of the homologous pair to exchange information. But then translocations are a little bit trickier because they can introduce DNA that wasn't supposed to be there. They can, break up some sort of gene pattern or introduce some sort of different diversity that's not really that good. Single gene disorders, I would throw this entire list on my cheat sheet somewhere. I think that it's definitely helpful to refer to because you might get a question that's like, what is a similar disorder to achondroplasia? Like, what's similar inheritance? And then you would just look on your cheat sheet and find them.
But we won't need to define them.
You won't need to define them, just know what's a similar inheritance pattern. Epigenetic modifications. So remember with Prader-Willi syndrome, that's a deletion of the father's part 15q. Angelman syndrome is a deletion of the mother's 15q. And so epigenetic modifications are when you methylate the DNA and you're like effectively silencing certain parts of your genome. And when you silence them, basically the next progeny won't express the gene that was silenced. So like, for example, like a certain protein or something that like would have been made won't get made or something to that effect. This example of edema formation I think is a useful conceptual idea. So like the idea of having plasma proteins, like when you have an oncotic pressure, think pulling towards you, so it draws fluid inward, and then hydrostatic pressure is something pushing outward. So I threw this on my cheat sheet just because to remember different scenarios of like if this had increased pressure, if this lost proteins, like what would happen next. So with edema formation, think, is the fluid accumulating or is it not? ADH secretion is kind of different than that in that ADH, remember, is anti-diuretic hormone. So if you anti-diuretic, so you don't secrete water anymore because you want to retain water. So if you have a low plasma osmolarity and you want to increase, or if you have a high plasma osmolarity, which means you don't have much fluid, you want to maintain more water in the system. So let's follow it here. So high plasma osmolarity or allality, doesn't matter, means you have low fluid volume. With low fluid volume, you want to secrete ADH because you want to stop excreting the water, and you want to retain water. You also stimulate thirst because you want to get more water into the system externally. So that eventually gets you more water in like the grand scheme. And when you have more water, you have more circulating fluid volume, which decreases the osmolarity, which will decrease, it'll feed back to the original, so you no longer are thirsty, you don't need that ADH secretion anymore. Renin, angiotensin, aldosterone system, this is also a really important one to know. So what happens is if you have a low blood pressure, you want to secrete aldosterone. In the final step, you want to secrete aldosterone because you want to retain fluid. So low blood pressure relates to low circulating fluid volume. So the kidneys secrete renin. After the kidneys secrete renin will activate angiotensinogen to angiotensin 1 in the blood vessel over here. Then angiotensin 1 gets converted to Ang2 by this ACE enzyme, angiotensin converting enzyme in the lungs. Ang2 can then act on the adrenal cortex. So the adrenal gland is sitting over the kidney. So you stimulate the cortex, which is the outer portion of the adrenal gland, and then that will secrete aldosterone. And remember, aldosterone is to hold on to sodium in the system. And if you hold on to sodium in the system, then you want to hold on to water in the system to kind of like dilute the sodium out. So think osmosis. So water goes to the areas of least water. So if you have a lot of solutes, so a lot of salts, a lot of, whatever, then that means you have less water proportionally. So then that water kind of flows into that area. Next we have atrial natriuretic peptide hormone. So this is kind of the opposite of the RAS system. So if you have a high blood pressure, you want to secrete this so that you get rid of sodium. So if you release sodium into the system, water kind of goes out. With potassium and calcium on membrane excitability, remember that these two determine sort of your electrophysiology, in that low potassium will decrease your excitability of the cell, but high potassium increases it because, remember, from the Nernst equation, that potassium is the ion that's controlling kind of the resting membrane potential. So if potassium goes up or down, that means you're closer or farther from the threshold. So if you have more potassium in the system, then you're more positive. And if you're more positive, you're closer to threshold, so you're more easily excited. On the other hand, you've got calcium, which determines what that threshold actually is. So if I have high calcium, I'm increasing the threshold, so it's harder for me to reach. And if it's harder for me to reach, less excitable. And then if I have low calcium, then it's the opposite effect. So it decreases the threshold, so it's easier to get excited. Next, we've got in the case of hypercalcemia versus hypo. So parathyroid hormone is kind of the opposite of calcitonin. So parathyroid hormone will try to increase the calcium content in your cells, or in your bloodstream. So if you don't have much calcium, so hypocalcemia, you release parathyroid hormone, which will stimulate the kidneys to reabsorb more of the calcium, and also you activate vitamin D. When you activate vitamin D, it will stimulate the gut, like the GI tract and intestines, to also reabsorb more. And if you also aren't getting enough calcium from these two, the kidney or the intestines, you can also break up the bone and you release calcium that way. Okay. On the other hand, if you have too much calcium in the system, you can decrease the parathyroid hormone, or you could increase calcitonin, which will pack the excess calcium into your bone and it'll kind of harden the bones. Okay? So they're kind of the opposites. Acid-base is a really important concept to get. You'll get at least 2 or 3 questions on this. So the whole idea of acid-base regulation is know your normal values. So pH is 7.4, and also CO2 is 40 millimeters of mercury, and the bicarbonate is 24. So most of the time I worry about the pH and the CO2 concentrations. So remember, pH is the negative log of H+, okay? So that means the more H+ that you have, the lower the pH is. And because it's a logarithmic scale, every one change, like change of 1 on the pH scale, is a 10 times change in H+ concentration. So on these clinical, like on the charts that he has, when it's like 7.4 to 7.3 or 7.2, that's actually still a pretty big difference. It's still like a 1 times like difference, moving 1 step in the H+ concentration. So then with CO2, remember CO2 can be converted into carbonic acid, which immediately will break up into H+ and HCO3-. So that so basically having more CO2 in the system is like having more H+ in the system, and more H+ means lower pH. Okay? And the same goes for the reverse. So less CO2, less H+, more higher pH. Either one of the charts that he provides, like that kind of goes over this, like I think are relevant for the cheat sheet. Let's see. I would have this equation with the pH equals the 6.1 plus log bicarb over the 0.03 times PCO2.
I have a question. Can you tell us for calculators?
Yeah, I was just gonna
Do you think?
I remember using a calculator, so I would bring it.
Okay,
Just bring one. Yeah, I'm pretty sure. Yeah, that we had to use this at some point.
And what's that equation for?
This is to calculate the pH if you know the bicarbonate and the pressure of the carbon dioxide. Yeah. So with this, like what you want to do is know the difference between respiratory and metabolic. So with respiratory, the way you read these lines is like the 40 is your normal CO2, and above that is your respiratory acidosis because you have too much CO2, and below that is the respiratory alkalosis, not enough CO2, so low H+ concentration, high pH. By the same token, like you got your, along the 40 line are your 2 metabolic problems. So if it's less than a pH of 7.4, it's acidosis, and above that is alkalosis, okay? So when I approach these cases that he gives you, or like diseases or conditions, first identify, is it acid or base? Also identify, is it respiratory or metabolic? And then you can determine, if you've narrowed it down to 2 that are like close to each other, then I would also consider, is there any compensation going on? So remember, if it's a metabolic problem, you compensate it with your respiratory. And then if it's a respiratory problem, you compensate it metabolically. So for example, if I had metabolic acidosis, so too much acid in the system, too much H+, then I have to get rid of the H+ by like breathing it out, right? So I breathe out carbon dioxide. So to get rid of carbon dioxide, that's hyperventilating. So then, yeah. So the CO2 concentration would be a little bit lower than 40. So definitely, if you're not comfortable reading these charts, practice. Yeah. So here's an example. So if I had prolonged vomiting, what does that do?
Decreases acid.
Right, so which letter is it going to be? So we've narrowed it down, so it's going to be like one of these guys, right? Because we're getting rid of acid. So is it A, which is kind of closer to 40, or is it E, which is closer to 20? A, because it's a metabolic problem and it's not respiratory related. So with chronic COPD, what about that? So COPD, chronic obstructive pulmonary disorder, so it's a breathing problem. So you're not breathing that much out. So if you're not breathing much out, then you're holding a lot of carbon dioxide in. A lot of carbon dioxide is a lot of H+. A lot of H+ is low pH. So we're going to be acidic, and then it's going to be a high CO2. So it's going to be letter B. Does that make sense for everyone? Okay. With this chart, I would have it on your cheat sheet in case he ever asks you, like, which one of these is like a coccus or gram-positive coccus that does whatever. Also note that in the original figure, he has mixed up where the cocci and the bacilli are. So if you copy straight from his PowerPoint, you don't have this correction of these like blue boxes. So make sure like on your cheat sheet that you know that like streptococcus is a coccus instead of a bacilli, because he has those reversed in the original file. Yeah, so these like, yeah, so with those blue boxes that he put in with the red arrow, that's correct. But on the original figure, if you just copy straight the figure, it's not going to show up. Yeah. Remember, staph is like the most common nosocomial infection. Clostridium, like Clostridium perfringens, can relate to gas gangrene. Neisseria is gonorrhea. Yeah. DNA and plasmids. Just remember that like bacteria are able to undergo conjugation to spread some sort of genetic material like horizontally to other bacteria. Viruses, remember, they are non-living organisms, and they're just packets of DNA or RNA that are encapsulated in some sort of protein envelope or protein protection. And they have to be infecting a host cell and use the host cell machinery in order to proliferate. So you can't have a virus replicate without a host. Overview of immune system. So we've got humoral versus cell-mediated. So remember, humoral is kind of like you encounter a pathogen like within the bloodstream, whereas cell-mediated is like the cell itself, inside the cell, intracellular, has a problem. And there's a difference between innate, which is more non-specific, and adaptive, which is very specific. So with phagocytosis, remember that there's pavementing in that the neutrophils, they kind of get up close to the site of damage. Then you have endothelial retraction. So these, like, the cells, they kind of loosen up a bit, and so it's more permeable, so more increased vascular permeability. Then afterwards you have diapedesis, which is when the cells can actually get through into the tissue. And then they go toward, the next part is chemotaxis, so actually getting towards the site of the wound where all of the chemoattractant factors are. Okay, so pavementing, endothelial retraction, diapedesis to get in, and then chemotaxis, getting towards that site of infection. Next we have with the T cells, remember T cells are made in the thymus. Everything else is made in the bone marrow for the immunity. And you've got different types of T cells, the T helper, T cytotoxic, and then you've got memory T, and then you've also got the memory B cells. So plasma cells secrete their receptors. So the B cell receptors, BCRs, soluble BCRs are the antibodies. Also remember, antibodies have they're like the Y-shaped. So the top parts are the variable regions. The bottom part is the constant region. And it's constant so that, like, let's say a macrophage could bind non-specifically to any antibody that's bound to an antigen and then engulf the whole thing. But the top part is what specifically binds to the antigen. E cells, IgE-mediated destruction of parasites. Remember, if you got a parasitic antigen floating around, it stimulates your B cell. Your B cell releases the antibodies, the IgE. The IgE binds to the antigen. A mast cell binds to the antigen. It'll release some sort of factors that cause the eosinophils to come into the cell. Eosinophil finds the parasite and degranulates and kills it. Yes?
So before the B cell secretes, right there, yeah. Before the B cell secretes the IgE,
Mhm.
does it become a plasma cell? Because that's a process that I'm not certain of. So
Yeah,
it's
so
like
like
antigen
a
and then-
B cell is like, it's like when you have a B cell, it can mature into a plasma cell, or it can become a memory B cell and just kind of be stored away. So the plasma cell is what's actually secreting antibodies, and it can like secrete many of them. So each of those B cell receptors, once it kind of detaches from the plasma cell, becomes the antibody that then binds. Yeah. Yes.
So like he said from IG something that you can make a class change.
Yes.
So when does that happen?
So most of the time it's going to be IgM that is like going to start off. So IgM, I don't have a picture of it, but it's like a pentamer, so it's got like 5 like little Y shapes all around, and that can become either A, G, or E. But then IgA is more in like the GI, the respiratory, the genitourinary tracts. IgG is the most prevalent. It's in the bloodstream kind of everywhere. And then IgE is like dealing with allergies, these parasitic worms. And then IgM is like the one that can do the class switch.
So then does that mean IgM? So then there must be a cell that secretes IgM, right?
Yes.
So then does the cell itself also change so that it secretes like IgA,
It's more like the M, like it's more like genetic than that. It's like the IgM has kind of like this DNA code that it kind of splices out other regions of it, and then like the antibody, like the IgM, will actually just change. Yeah, but you don't need to know about that. Yeah. So you've got MHCs and T cell receptors. So T cell receptors, remember, they don't float around in the system, and they're not like B cell receptors. So B cell receptors are your antibodies, but you don't have like a T cell antibody. So remember, with T cells, they have to have the major histocompatibility complex and the antigen presenting cell present the antigen to them. Okay, so antigen presenting cells can be B cells, they can be dendritic cells, they can be macrophages, and they, attached with their MHC, can present an antigen to the T cell. Also note that with the T cells, it's also, the MHCs are also called HLA, human leukocyte antigen, and then MHC class 1 is more for like self antigens, and that deals with CD8 or cytotoxic T cells, whereas MHC class 2 deals with foreign, and that's CD4 with T helper cells. Okay, so it's a little bit different what they do. So MHC class 1, self, with CD8 T cell cytotoxic. And then MHC2, class 2, is with the foreign and the CD4 with the T helper. So I remember it's like 1 times 8 is 8, 2 times 4 is 8 as well. Hypersensitivities, I throw this chart on instead of this one, and I also take this like bottom section if I feel the need to. I'm not really sure if I threw. I'm pretty sure I threw this on though, rather than this one. So, because this one kind of tells you everything that you kind of need to know. So like which disease fits into which category. So with type 1 IgE mediated, remember that's more allergies, type 1 allergies. Type 2 is tissue specific, so like your immune system is reacting against yourself. Type 3 is immune complexing, so that can be with lupus. And remember, with lupus, it can also be due to, it's like a disease that's caused by environmental triggers, and I don't think that's like emphasized that well. I don't know if I have a slide of that. Yeah, so lupus with environmental triggers, you might see that written into the question, and usually that's kind of glossed over. But it's like your cells are binding against your DNA rather than just proteins. Normally antibodies bind to like protein antigens, but in this case it's more like the DNA, which is special in that case. So type 3 are immune complexes, so you get these aggregates of like antibodies and like their antigens all bound together, and it kind of gets detached and precipitates out into your bloodstream, and then your body starts attacking that huge aggregate, and then that causes some sort of immune response. T-cell mediated is probably the one that's like least understandable, like conceptually. Like there's just certain cases that like are more common, like Hashimoto's, thyroiditis, and type 1 diabetes. And remember, T-cell mediated is the cell itself has a problem. It could be like virus infected or something like that. But I would just throw this sheet on, and then just remember like type 1 IgE, type 2 and 3 are IgG, and then type 4 are all your T cells, which is pretty straightforward to memorize. So we just kind of went over all of these. With blood types, remember, if you have type A blood, that means you have A antigen and anti-B antibody. So with type AB, you have AB antigen, but no antibodies. And then type O, you have no antigen, but you have anti-A, anti-B antibodies. Okay? So with type O, you can donate that to other people because their body's antibodies won't react with your blood. But if you donate too much type O, then your antibodies could bind to their blood. Does that make sense? And then with type AB, it's a recipient because it doesn't have antibodies, so it won't attack. But their antibodies eventually could attack you if you had too much blood. So AB is typically the recipient, and then O is typically like the donor, universal donor. So with these immunodeficiencies, I also threw like a list of these on my cheat sheet, just in case you forget. So with the SCID, the severe combined immunodeficiency, that's when you don't have B or T cells, but you can still have your like innate immune system. But these people are very immune deficient, and they have to be kept like in a sterile environment. Like think the bubble boy. A gamma globulinemia, that's when you don't have B cells, but you still have T cells. And then DeGeorge, that's when you have no thymus, so you don't have T cells, but you do have B cells. Okay? Chronic granulomatous disease, so you can't produce reactive oxygen species to kill bacteria, so your macrophage could like engulf the bacteria, but then it's not actually able to kill it, so bacteria can kill your cells. And then HIV, remember, HIV is an RNA virus, so it has to be converted into DNA before it can incorporate into your genome and replicate, okay? So you could theoretically have an entrance inhibitor, so preventing the virus from coming in. You could have a reverse transcriptase inhibitor to prevent the RNA from converting to DNA. An integrase inhibitor to prevent the DNA from getting into the nucleus where your genetic material is. Or you could have this protease inhibitor that prevents proteolytic modification. So certain proteins, like once they're made, they have to get cut up in certain places so that they can be active. So in this case, like if you prevented that cutting up, then it's inactive. Moving on to cancer. So we've got proto-oncogenes and tumor suppressor genes. So remember, proto-oncogenes are all about preventing. So one is bad enough to become cancerous. So oncogene is a cancer. Proto is just like the step before becoming cancerous gene. Tumor suppressor genes, on the other hand, are more like your guardians of your DNA checkpoints. So if you knock one out, you still have the other. But you have to knock both out for it to become bad. Proto-oncogenes, I would throw this list on. You don't have to know what each one specifically does, but just know that these are all examples of proto-oncogenes. So one can become one defective is bad enough. Translocations can be especially dangerous if you combine like two transcriptional factors, like one thing, like things that aren't supposed to go together, or one that could like constantly turn the other thing on, and then you overproduce. So either way, chromosomal translocations can become cancerous or just bad in general. Remember, non-homologous but balanced translocations are translation. Tumor suppressor genes, again, throw the list onto your cheat sheet, and we'll go more in depth into specifically P53 and retinoblastoma 1. So RB1 are the ones that like he goes more in depth into. Remember that there's 6 hallmarks of cancer. Now it's actually 10, but we just focus on these 6. So with self-sufficiency and growth signals, RAS. So RAS is a protein responsible in this system to turn this signal off. So if you turn the on signal, and if you prevent the on signal, okay, let's start over. So it's GPCR, okay, which is the G protein coupled receptor. So growth factor binds to the GPCR, and it causes the these inactive complexes to kind of change conformations, whatever, and you swap out GDP for GTP. So GTP will make it active, and then the RAS will convert GTP back into GDP, so it'll turn the system off once you don't want any more growth. So if I cut out RAS, which is what turns off the growth, then that means I'm constantly on. Okay, does that make sense? So I leave the off switch permanently like in place so that I'm constantly growing. So that's why it's self-sufficiency and growth signals. And so it's important to note, like with the 6 hallmarks of cancer, like to know what his protein examples or whatever are. So in this case, self-sufficiency and growth signals, RAS. That'll be like, one of your quiz questions or whatever. Insensitivity to growth, anti-growth signals, so retinoblastoma. So retinoblastoma is one of your tumor suppressor genes. It's like a part of the checkpoint. So if it prevents like the cell from growing if you're not ready to grow, but if you knock it out, basically, then like the cell can grow. So insensitivity to anti-growth signals, retinoblastoma. Does that make sense? If you have a sporadic mutation that's like one by chance, it's very, very unlikely that you'll get another one. But if you're born with one, so familial, you'll basically have like 100% chance of getting that second bad mutation. Evading apoptosis, so P53 is another guardian of the checkpoint. So P53 protects the DNA, so if you have DNA that is not ready to move on, or it's somehow cancerous or DNA damaged, P53 will either start the cell like to undergo repair, or it'll tell the cell to go undergo apoptosis and just kill itself. But if you knocked out P53, then cells can evade apoptosis. They can just keep replicating no matter what. Like they won't respond to these death signals. Next, we've got limitless replicative potential with telomerase. So with telomerase, remember, it's extending the telomeres. So in normal cells, we have a limited length of chromosomes because of the telomeres. So like once the telomeres shorten all the way down, you can't replicate anymore. But with cancer, they like increase that length again so that they can always keep replicating. Sustained angiogenesis. So VEGF is vascular endothelial growth factor. So cancer needs tons of nutrients, so it needs blood supply. So it stimulates these like new blood vessel formation to like attach to it and give it nutrients, give it blood, give it your oxygen, give it whatever. So VEGF. Metastasis, remember, is like when you've got a primary tumor and eventually it breaks into the bloodstream. Once it breaks into the bloodstream, it can get all to different places. So most of the time it hits the lungs or the liver, but with the bone can be hit with like breast and prostate cancers. That's just another example. But like most of the time you get the liver and the lungs getting hit because that they are constantly in contact with blood and they're constantly circulating basically everything that goes through the blood system. HPV is a virus that's known to cause cervical cancer, but it also causes warts, genital warts, foot warts, hand warts. But remember, warts are not cancerous, but HPV can cause cervical cancer. So it does that by having 2 proteins, E6 and E7. So E7, so early protein 7, can neutralize retinoblastoma protein, so you don't have the checkpoints being manned anymore. And then if you have E6, it can bind to P53, and you evade apoptosis. Okay. So that's pretty much it for the cancer section. Also, for the cancer section, it's good to have the breakdown of like the what percentage of each cancer was like in the male and female, like which is the most common, that thing. There's like one of the very first slides with like the people on it. I would record those numbers down just in case like he asks like what is the most common like prostate cancer or like whatever. It's good to have. So with endocrine, just remember the whole idea is feedback loops, feedback mechanisms. So the brain is like your primary, control center, telling the body like, we need this hormone, we need this whatever. The brain will tell, some sort of secondary organ or whatever to pass on, or tell a third organ to do something else. So most of the time these signals originate from the anterior or posterior pituitary, and that's in the brain. So, and then from there, they stimulate different organs to secrete the corresponding hormone. So with the anterior pituitary, you secrete flat pig, so FSH, LH, ACTH, TSH, prolactin, and GH, growth hormone. Whereas the posterior only does 2. So that's ADH and oxytocin. And we'll go more in depth. So to reiterate, the anterior pituitary secretes all of these. They stimulate a target hormone. Target hormone will then secrete the corresponding next thing. So I would just read this over. Try to orient yourself. So TSH, thyroid stimulating hormone, stimulates the thyroid to make T3, T4. ACTH, adrenal cortex to make cortisol. FSH, LH stimulates ovaries, testes for secondary sex characteristics with like estrogen, testosterone, that kind of thing. Growth hormone can stimulate the liver to stimulate insulin growth factor, that's the IGF, or tissues directly. It can tell the tissues to grow themselves. And then prolactin is secreted by the breast for milk production. So with thyroid hormone, T3, T4, so T4 is the most prevalent, but T3 is the more active form. This is for growth and general metabolism in the cells, okay? So if you don't have a good thyroid, then you can have a lot of metabolic problems or growth issues. And so remember, feedback loops. So 1, the hypothalamus tells the anterior pituitary, okay, we need XYZ. Anterior pituitary tells the target hormone to secrete whatever. And if the target hormone actually does secrete whatever, then it'll feedback negatively on both the hypothalamus and the anterior pituitary to stop sending signals to secrete whatever it's secreting. Parathyroid hormone we went over already. So with the adrenal gland, remember it's got an outer cortex, these 3 outer layers, glomerulosa, fasciculata, reticularis, and an inner medulla. So the 3 layers, zona, whatever, those are all part of the cortex, the outer part. Medulla is the inner part. So the glomerulosa secretes the aldosterone. And remember, aldosterone is for sodium retention. And if you retain sodium, then you retain water. So that's for like the RAS system, RAS, renin-angiotensin aldosterone system. And then the zona fasciculata, you've got cortisol, which is like for glucose metabolism and like stress reduction and anti-inflammation, that kind of thing. And then reticularis, that's for dehydroepiandrosterone. DHEA, so like basically weak androgens, and that's for like secondary sex characteristics. And then the medulla is your catecholamines, which are epinephrine, norepinephrine. So think like adrenaline, like getting yourself hyped, that kind of thing. So aldosterone, renin-angiotensin system. I would know this chart. I don't know if you necessarily need to put it on your chart, but things that can stimulate aldosterone, remember hyperkalemia, because if you activate aldosterone, you retain sodium, but you secrete out the potassium. Okay, so if you have too much potassium, you can also make aldosterone like get rid of the potassium by like holding on to sodium. It's kind of like the way the kidneys actually work. Hyponatremia, so not enough sodium in the system, you want to retain it. Or ACTH, adrenocorticotropic hormone, like so stimulate the adrenal gland to actually like secrete whatever it secretes, and it secretes aldosterone. So cortisol, anti-inflammation, growth suppressing effects, influences awareness, sleep habits, carbohydrate metabolism. So cortisol stimulated by ACTH. Again, another feedback loop. So with these lists, I threw them on my cheat sheet just because there can be a lot of words and you might not remember like immediately like, oh, this is a, too much or too little. So like this might, it's not for everyone to put on their cheat sheet, but if like you have trouble remembering all the different things, like it's worth looking into like, can you squeeze it on, like just a list of like what they all do, and then you can write in yourself any little notes that'll help you remember what each of these are. So remember with the posterior pituitary, we've got diabetes insipidus. So posterior pituitary, remember, secretes two things, ADH and oxytocin. So if you have too little ADH, then you're not holding on to fluid as easily. And if you don't hold on to fluid, it's all going to go into urine. So polyuria and polydipsia for thirst because you're not retaining any of that water. Urine will be hypoosmotic because so much fluid is diluting out whatever solute there is. And then your blood becomes hyperosmotic because you don't have any fluid to dilute all like the concentrated part inside the system. So the urine gets diluted, hypoosmotic. The blood is hyperosmotic. It's not getting diluted. A neurogenic effect is due to the posterior pituitary. So neuro, think brain. So the brain is not secreting the ADH versus nephrogenic, kidney problem. So you secreted the ADH, but the kidney is not responding to it. So neuro versus nephrogenic. SIADH, that's syndrome of inappropriate antidiuretic hormone secretion. I think this is one of your clinical cases. So if you have too much ADH, you're retaining too much. And if you retain too much, your urine is hyperosmotic and you have all these other problems with like water retention and that kind of thing. Hypopituitarianism. So you've got, again, like all these different things. Like, again, you can see whether or not you want to make note of it on your cheat sheet, but it's the same idea. So hyperpituitarianism, you're not secreting, each of the flat pig hormones. Hyper, again, so if you're secreting too much, you can have visual changes, different. You could have a null cell adenoma that doesn't actually secrete anything, but it takes up space in the brain. Excuse me. So cancers in general in the brain are very dangerous. They can edge out kind of like the corresponding area for like the other parts of the brain to, have space. You could have this bilateral hemianopsia where like depending on if you got like a stroke or something, like sometimes you lose vision in different areas. And so depending on where you lost the vision, you might have left, right, or, outer portion blindness, like depending on how things cut off. So if, remember your eyes, like everything in the brain kind of crosses. So like cutting off like right side eye connection kind of affects like the left side of the brain and et cetera, et cetera. Let's see. So if you have too much growth hormone, acromegaly versus gigantism. So acromegaly is during adulthood. Gigantism is for kids. And then hyposecretion is pituitary dwarfism for kids. And that's another example of dwarfs. Whereas remember, achondroplasia was a genetic dominant, autosomal dominant disorder that led to dwarfism. So this is purely endocrine, completely different than genetic. And then aches and pains if it's like with age. Sure. Okay, prolactin. Prolactin is for like milk production. So then if you produce too much, then it can cause different syndromes in females, like amenorrhea, so like not having a regular period, hirsutism, osteopenia, so weak bones. And then in males, you can have like problems with like sex function. Thyroid pathologies, hyperthyroidism, so you could have Graves' disease. Again, like you can throw all these on your cheat sheet. And don't forget that there are these 4 classifications, like papillary, follicular, medullary, anaplastic. I would just read them over, make sure papillary is like most of them, follicular is some of them, and then anaplastic is the rarest. Feedback mechanisms, I threw this chart onto my cheat sheet as well, just because I felt like it was necessary. Also to have like where it all fits in. As for symptoms, it's good to just read them over and try to understand, like, do I know why this person is anxious? Do I know why this person has, like, whatever? Treatments you don't have to know, so for Graves' disease, hyperthyroidism, so too much metabolism kind of stuff. Hypothyroidism, not enough metabolism, not enough, like, dealing with different things. So you could have like the elevated blood cholesterol, myxedemia, puffy face. Hashimoto's, remember, is a type 4 hypersensitivity. So it will attack your receptors for the cells. Iodine deficiency, so like if you don't have enough iodine, that helps to make like the T3, T4. So not enough like metabolism, and you get these kind of manifestations. Again, I would throw this kind of chart onto my cheat sheet, and instead of like writing out elevated versus decreased or whatever, you can put up or down arrows, shorthand it basically, save yourself some trouble. Let's see, parathyroid pathology. So again, it's like if you have too much parathyroid hormone, think, what does parathyroid hormone do? Deals with calcium, and then you can kind of get it from there. Adrenal cortex pathologies, you can have hypercorticism, like Cushing's, hyperaldosteronism, or adrenal cortex syndrome. So with Cushing's, remember there's 4 different causes. With Cushing's, you could stimulate the cortex, and if you stimulate the cortex, then you're not getting. If you stimulate the cortex, like from the ACTH, the tumor is in like the source of the anterior pituitary. So too much ACTH, and even if you produce the cortisol, it won't stop the feedback mechanism. Whereas if the tumor is in the adrenal cortex itself, then like you will have the feedback, like you'll tell the anterior pituitary to stop, so you don't have much ACTH, but you have a lot of cortisol no matter what. So this is like, so the first case is a lot of ACTH, a lot of cortisol. The adrenal Cushing's syndrome here is not much ACTH, but a lot of cortisol. The paraneoplastic is like, you get ACTH, but it's secreted from like an ectopic, so the an ectopic cancer, so the cancer is like in the lungs, let's say, and it tells the cortisol to secrete. Or you could have iatrogenic, so that's caused by a drug, caused by a doctor. So again, looking at the symptoms, everything. Hyperaldosteronism, so that's Conn syndrome. So this is good to know that like you have the tumor, secretes aldosterone, you hold on to a bunch of sodium, so you don't really stimulate the renin system anymore. Let's see. With adrenogenital syndrome, it's a problem with this like 21 hydroxylase deficiency, and you can have like virilization, so that's like problems with secondary sex characteristics. So like, and then adrenocortical insufficiency, so Addison, so you have decreased aldosterone, cortisol, weak androgens, like, so it's kind of all around insufficiency for Addison's. And lastly, the adrenal medulla pathology, it's this thing called pheochromocytoma. So you have a cancer of the chromaffin cells, which are the ones secreting like adrenaline. So you have like super high adrenaline levels, and you can get really spooked really easily, and it's very dangerous. I think that's it.
Thank you.